since everyone's gone quiet, I'll start my introduction, I guess. So it is my pleasure today to introduce Cassandra Donatelli. So Cassandra took her BA from the University of Rochester in a program called Archaeology, Technology, and Historical Structures, which is a spin-off of their mechanical engineering department, and where, looking back at her CV, she apparently did a project on, among other things, continuous fermentation of wine, which she put on her CV. I met her in the fall of 2013. So we didn't really have a formal visitation procedure at that point for graduate students, so she came in, said she was interested in the lab, her CV, it was all great, we accepted her right away, and then that was all good, and then in March, I got another email, I had another conversation. She said, hey, I'd like to go out to Friday Harbor again this summer, and can you pay for it?
I don't think I have to quite like that.
Well, she might not say it, but basically that was the gist of it. And I was a new assistant professor, and I just recently gotten a grant, and I had sort of more money than I knew what to do with, so I said, sure. And if I know Cassandra, she probably said something like, it'll be great, I promise. And it was. So she went, and she spent the summer at Friday Harbor, and she came back with the core of what you'll see today. That became her thesis. That was really the beginning of it, even before she started here. And so she kept her promise. And that's been kind of the problem with Cassandra all the way through, is when she says she's going to do something, she does, and she does it well. And so she's done a lot, and a lot of people have asked her to do things. So one thing I wanted to do as part of this introduction is just tell you a few of the things that aren't in here, just so you can get some idea of the breadth of the different things she's worked on while she's been here. So she was important in the construction and design of a Caterpillar robot, an award-winning Caterpillar robot, which is not related to fish at all. She was instrumental in a project for measuring the punching force of mantis shrimps, which we had published not too long ago. She's also been involved in another project estimating forces on the pectoral fins of sharks as they're turning. And she was a co-author on, I think, 6 different abstracts at the SIGP conference in January, only 2 of which I think actually came from my lab. One of those was a project studying the skeleton of starfish, and using fractal dimensions to figure out sort of the complexity of different starfish. She's also been working recently on a project to untwist fish that got twisted in a CT scan. So she's worked on a whole bunch of different things, covered an awful lot of bases, but today she's going to tell you about the core of her dissertation, which is called The Role of Mechanics in Elongate Fish Swimming Behavior, Comparative Studies of the Vertebrae, Skin, and Body in Sinking Fish. But before she does that, just to keep her company, an emotional support eel.
Very important. Emotional support eel. All right. Hi guys. Thank you all for coming today. I'm really excited to see so many of my friends here in the audience. So I hope I can keep you entertained for the next 45 minutes of your lives. And I'm going to do that by talking about fish. Shocker, I know. So I really like fish, and I like fish because they're so diverse. They come in lots of different shapes and sizes, from elongate eels like this, all the way up to box-shaped fish like the one at the top of the screen, and pretty much everything in between. They're really, really diverse in terms of their body shape. Along with their body shape is the way they swim, and we can pretty much say that the number of fish species that exist, there's that many different ways to swim. All fish swim just a little bit differently, and they come up with some really cool ways to get through the water. One of my favorite ones, I don't know how well you can see it, is the ribbon eel right here, and it swims by passing a lot of waves on its body. It's very cool, that's why I like it. But other fish use just their pectoral fins, they're flat through the water. Some have these tiny little ribbon fins on their back. Some have really stupid, modified dorsal and anal fins like that sunfish up there. It only barely makes it through the water. But that leads us to the question of how can we compare morphology and behavior in these fishes. They're all so super different. They swim really different, they're shaped really different. These two fish are from a similar area. They're both from Friday Harbor. This is a gunnel, so it's elongate, bendy, slippery, it doesn't have a lot of scales. And then this is a sculpin. It's got a really big, crusty head, and it's grumpy all the time. So how can we compare these two fish, even though they're so different? And one way that people have been doing that is looking at vertebral morphology or skeletal morphology. One of the very few things that all fish have in common is having a skeleton. It's what makes them vertebrates. So it's something that all fish have that we can look at. And we've been looking at it in a lot of different ways. So one of the original ways of looking at skeletons was just letting the animal die and letting it dry out and gluing it back together again. You can take x-rays, which is just a snapshot through the animal. Clearing and staining is a really nice way to keep the fish kind of whole in 3D, and you dissolve all the muscle off and stain the bones pink. And finally, we have these fossilized fishes. You can see their bones preserved over time as well. One of the newest fun ways to look at skeletons is using micro CT scans. So this is a CT scan of one of my gunnels. I will stop it from spinning so you don't all get sick. And it's one of my favorite scans. I just love how the vertebrae are perfectly repeated down the length of the body. Just to orient you a little bit, this side is the head. For those of you who aren't familiar with looking at fish, this is the tail. All of this stuff over here is gut contents, so whatever it ate before it died. And this is a radio-opaque marker that we use to identify the fish in the tube of scans. We scan like several fishes in one go, so the only way to identify them afterwards is to put a marker. And we've kind of got our work cut out for us with these fishes. There are 30,000 species of them, more than that actually. And if we're trying to answer questions about all fish do this, or all fish behave this way, or have this morphology, or have this shape, we have a lot to look at. The fish that I've been looking at fall all the way at the bottom of the tree, way down here in this little group. I looked at 16 different species in that group. If you spread it out, this is kind of what I've looked at. I tried to get away with this tree, but my lab wouldn't let me. They wanted me to show you that my little more fish tab at the top there meant all of this. Thought it was unfair for me to cut it out from the tree. Fair enough. That's what I mean by more fish. And also, just to give you some context where you are, you're at the top there in tetrapods, and then sharks are all the way at the very top of this tree. So, tree of kind of life, of fish life. There's lots of different species to look at. We have a lot of work to do if we're trying to compare everybody. In addition to just answering questions about evolution, looking at how animals swim and the way things move has actually been really important recently in the field of bio-inspired robotics. It's actually starting to become a thing. I used to kind of make fun of it and being like, they're not really doing biology. But now there actually are. There's like a lot of roboticists that do real biology, and we actually work together with roboticists to make things that can help both them and us. This is a GIF of the robot that Eric was talking about before, caterpillar robot. I think it's cute. It's just rolling over. And these are some other robots that we use to study biology thanks to working with engineers. This one is teaching us how neurocontrol leads to motion. These two, the one at the top and over there, are environmental monitoring robots, so they swim around in fish's natural environments and tell us things about the way the world works without us there bothering them. We can even use them, like this one here, to study extinct animals. So that robot was made to teach us how we learned to crawl out of the water onto land way back in the olden days. And speaking of dead old things, fossils. Another good reason to try to make patterns in morphology for all the different species of fish that I showed you before is that there's a lot of things that are no longer alive. There's a lot of fossils, a lot of extinct animals. And if we have some idea of how morphology leads to function, we can then make predictions about how these animals might have behaved or where they might have lived millions of years ago, which is probably not correct, probably hundreds of millions of years ago, but I'm not a fossil person. how fish swim. I've done the introduction. I'm going to be looking at how their body mechanics affect their swimming. I'm going to skip these two sections about skin and some technology things there. And we're going to jump to the bottom and look at how the vertebral morphology affects body swimming and mechanics. So let's start off with how fish swim. Looking at fish swimming, I picked 6 different species, which some of you may be familiar with. These are 6 elongate fishes from Friday Harbor. You'll see a trend here. Most of my fishes are from Friday Harbor. And you might also catch that I've been talking about fish diversity and fish morphology and how different they all are, but I picked 6 species of elongate fishes that all look kind of the same. And the reason I did that is you have to start somewhere, and they all do a really cool kinematic thing that I'll show you in a second. Also, they are different in one kind of important way. They're from different habitats. So the first 2 over there are from the intertidal zone, and that's just the area where it's where you walk around on the beach, where you'll find tide pools, you can flip rocks and catch crabs. Those fish will end up in those tide pools and under those rocks. If you look all the way at this other end, the subtidal fish, they live way past the beach. So they're staying on the bottom of the ocean, way past where everybody else would be. They don't really have to worry about the tide coming in and out. And these nearshore fish do something a little bit different. They follow the tide in and out, so they don't usually end up in the intertidal zone, but they also are constantly moving. As the tide goes in and out, so do they. So the thing I am interested in with these elongate fishes is a body twisting wave, which we call wobble. And if you look at this video of a fish swimming by, this is a gunnel. Most of my videos are gunnels, they're my favorite. And you imagine taking a freeze frame of it and then taking cross sections of his body along the length. You can see that his tail and body rotates and twists along the axis as it's swimming. And this is pretty prevalent in this fish and the other elongate species that I showed you before. And this twisting along the body is what we call wobble. And you may be wondering, why fish wobble? Why is this an interesting parameter to look at? It's just twisting. How interesting could it be? Well, it's interesting because all the fish that I study, along with this fish that I don't study, but it's very cool, this green eel, they're negatively buoyant fish that sink to the bottom of the ocean when they're not swimming. But they do need to swim back and forth. Like I said, the nearshore fish are always swimming back and forth following the tide. So dragging their bodies along the bottom would be a pretty inefficient way to go. So what I think they're doing is I think they're wobbling to create lift up and down with their bodies so they can push up off of the bottom of the ground and swim nicely. So this eel is trying to get away from this diver, and you can see that his body is wobbling pretty extremely from left to right, which is letting him swim up off the bottom and away from the annoying diver. So I measured that twisting in these 6 species here, and we found a pretty fun way to do that saved us a lot of time in digitizing. For those of you that study kinematics or movement, you know that the worst part is clicking points on a screen to digitize every single thing it does. But we actually wrote some software to do it for us. So the software goes through the video like you can see here and takes frame by frame, takes the outline of the fish, traces the midline, and then spits out a bunch of parameters that we're interested in. So from the midline, we get all your standard 2D swimming parameters, like tail beat amplitude, which is just how much the tail goes back and forth. We can look at all the wave parameters, see how much the fish bends along the length of the body. And we can also use these traces to measure wobble. And we can do that because we know the shape of the fish. We know how tall the fish is, and we know how wide the fish is. So if we look at the traces and we can see, for example, if you look at this teal one right there, you can see that it's really, really narrow. And if you look at the purple one, you see that it's really wide at the tail. So the difference between really, really narrow and really, really wide corresponds to wobble. And just to put that in schematic form, if you see a really, really narrow section of the tail, it's wobbling 0, it's not twisting at all. And if you see a really wide section of the tail, it's twisting or wobbling a lot. So that's how our computer program tells us wobble in all of the fishes. And once we've measured it, we can look at it over time, like this. So I'm showing you tail beat cycle on the bottom here, so this is over time, of course with one trial. And I'm showing you wobble over on the y-axis. And for one species of fish, the gunnel that I've been showing you all along, this is what the wobble wave looks like. It's actually a nice, predictable wave. And if we look at all the other species as well, there's some wavy things happening. So all of them have some sort of wave that goes on while they're swimming, but it's very clear that they're different. So this green line is a lot further down than this orange line. They all wobble, but they all wobble differently. So to better understand how the different fish wobble while they're swimming, we took the max peaks of all these trials, over all the species, over all the individuals, and we calculated the max. So now I'm going to show you position along the body, and this is going to be really common throughout the rest of the talk. The x-axis, if it's position along the body, 0 is the head, 1 is the tail. I'll try to repeat that, but it's a thing that you'll see a lot. And in this case, the y-axis is max wobble. Alright, so for all of the fish, if we plot wobble along the length of the body, we get these lines here. You can see right off the bat, they're all over the place. There's not just one pattern that all of the fish do. So even though they all swim kind of the same when you look at them in 2D, when you look at their 3D swimming, it's pretty different. And what we found from this was that if you compared species from the same habitats, those habitats that I showed you early on, that's when we started to see some patterns come out. So fish from the same habitats tended to wobble in the same way, which is pretty cool to find. So I've been telling you a little bit about wobble and how I think it might produce lift, and I just wanted to briefly go over some evidence for wobble producing lift. And to do that, I need you guys to start thinking about the end of the fish's tail as a wing. So the end of our fish's tail here is really flat, shaped kind of like an oval, very similar to this fly wing. And there's been a lot of studies on flight. People really like flight. So if we compare fish swimming to flight, you can start to get some idea of what the fish might be doing. So if you take a cross-section of the fish tail and a cross-section of the insect wing, they kind of look similar. The insect wing is going to be a lot flatter, like you see here. And this little green oval is the fish tail. It's a little bit wider. But generally, both wing-shaped. And this graph represents a study from an insect wing biologist. And what they found was that they looked at angle of attack. So in flight, you call the angle of the wing angle of attack, which directly corresponds to wobble if we're thinking of our tail as a wing. So these angles of attack correspond to this amount of wobble, just for your reference. And what they found was that for certain angles of attack, insect wings produce the most lift right here in the middle, around between 36 and 54 degrees. The lift coefficient was highest, which means that the lift they're producing is the most at that point. And if you compare that to the way our fish swim, the thing I'm showing you over here on the right is a video looking behind the eel. So imagine an eel swimming through the screen, and this is the cross-section of his tail. We can take videos of that, which I'm going to attempt to play for you. And they're high-speed videos, so it's playing back in very slow motion right now. But if we take these videos, trace them, and then play them back at slightly higher speed, I hope you can see there's a green oval rotating across the screen. And it turns out that the angle of that oval going side to side matches pretty well with the highest lift coefficients in insect wings. And to put that another way, if we look at the wobble graph that I showed you before, this area is the area where they found the highest lift coefficient in insect flight, and you can see that a lot of the species fall right within that range. So if these species are attempting to produce lift, they're doing it right. They're angling their tails at the way that would do it best. We also have a little bit of hydrodynamic evidence that leads towards us thinking that tails produce lift. So again, this video that I showed you before is a video from a PIV study, and PIV stands for particle image velocimetry, which is a fancy way to say we put particles in the water, light them up with a laser, and do math on them. So all the little sparkly dots you see here are tracer particles. They're lit up in a light sheet right behind the eel's tail like this. The water is flowing out towards you, and the eel is swimming against the water in towards the screen away from you. So if we take videos like this of the eel swimming, and we put them through and do all the math on them, we get results that look something like this. The important takeaway here, so I'm showing you vorticity, which in short is how the water is rotating. So if you see counterclockwise vorticity, the water is spinning counterclockwise, and if you see clockwise vorticity, the water is spinning clockwise. I would draw it out for you, but I'm just going to do it backwards, so look at the arrows. So what you can see here in just one snapshot of a eel tail is that there's different types of vorticity at the top and bottom of the tail. And if we put that in schematic form so that it's not as painful to look at, we can see that as the tail moves, I'm going to show you some snapshots of the tail moving across the screen this way. It starts out with clockwise on the top and counterclockwise on the bottom, and then as it starts to beat across the screen, the vorticity changes, and by the time it gets to the end, it's got these two smaller vortexes of water at the other end, which could be producing a jet of force up, giving it the lift that it would need to swim up off the bottom. I know I'm really not a big fan of in the future I plan to do this, but in the future, this summer, I plan to go into this data a little bit more so I can give you some more on it. It's pretty exciting and I'm excited to work on it. But not for today. For today, we're moving on. And we just talked about how fish swim. And I want to ask the question, do the fish wobble on purpose? So I've shown you that they do this crazy weird twisty thing. But are they doing it on purpose, or are they just passively wobbling? If they're really, really flexible, you might imagine that they would just wobble more from being flexible. And if they're really stiff, they might wobble less. So to answer that question, we looked at the mechanics of their body and compared that to the amount of wobble. The thing we measured, among other things that I'm going to tell you about today, is torsional modulus. And that's just basically a measure of how much force it takes to twist an object. And since wobble is a twisting wave, we measure fish twisting. You can see here is my fish twisting rig with a dead fish that we are twisting to measure torsional modulus. And torsional modulus, in short, is basically just the force to rotate over the shape of the body. So all the fish are elongate, but there's slightly different in shape along the line. So to account for the slight differences, we use this so that we can look at the modulus, so we can look at the shape of their body. I was going to say no fish were harmed in the making of this, but they were already dead. Just go with that. All right. So again, I'm showing you the same wobble graph over on the other side, which you've been seeing before, showing wobble along the length of the body for all the fishes. And if wobble was passive, and if wobble depended on the mechanics of the body, we might imagine a fish that wobbles a lot at the tail would be really not stiff at the tail, and vice versa. So that's what we would predict if wobble was a passive thing. What we actually found was that all these fish tend to have a pretty similar trend of torsional modulus from head to tail. They all start off not so stiff and then get stiffer, which doesn't support the hypothesis that fish wobble is related to fish body mechanics. It's clearly not. They look very different. So that led us to think that something else might be going on, but it also means that we have this great data set of fish swimming and fish body mechanics, which lets us start to put things together and look at how the vertebral column and other morphology might be a more important role in measuring these things. So now we're going to move on to vertebral morphology and see if we can use the shape of the vertebral column to look at the swimming things that I showed you before. And just to give you another view of my lovely fish scan here, the part of the fish that we're looking at is the vertebral column, this long part that takes up most of the fish's body. These fish are basically a muscle-wrapped vertebral column, easy and perfect. The specific part of the vertebral column we're looking at is just the inner bit of the joints. So if you take two vertebrae here, well actually I should pass these out, give me a second. You take two vertebrae here, and if you imagine cutting it down the middle this way, that's what it looks like on the inside. It's an hourglass shape. You see here, the hourglass. And the things that we measured all had to do with what this shape looks like on the inside for different fish. So we measured the cone diameter. The cone diameter is this guy right here. We measured the canal diameter, which is this little bit here. We measured the angle of that cone, and we also measured the length of the vertebral body. And the reason we chose to look at just this specific part of the vertebrae when there are so many other things to look at, you can see they've got spines and processes and all sorts of other things going on. But we chose to look at the inside bit, mostly having to do with how fish develop. And there are probably a couple developmental biologists in the room right now, so feel free to correct me if I say this wrong, or you know what, just go talk to them later, they'll tell you how it actually works. So fish go through this complicated metamorphosis life history where they start off as little larvae and pretty much completely change into being adults. When they start off, their vertebral column isn't a vertebral column at all, it's just a notochord that wraps around their body. Similar to all of us, we all start out with a notochord as well. And that's just a kind of bendy yet stiff rod that's just in the body when they're little fishies in eggs. When they hatch out and start swimming around in the water column, they start to develop these little hard bits here. So this red represents bony bits. And as they start swimming around, these bony bits start to ossify, which means get stiffer and turn into actual bone instead of just flexible notochord tissue. And by the time they're adults, or in this case juveniles, you can see that we have that really nice hourglass shape that I showed you before. And all fish go through this process as they're developing, but the amount of inside bit that they keep is different for different species. So there's always a bit of notochordal tissue and connective tissue. It's stuff that started when they were tiny little fishies in eggs that they retain throughout their lives. And looking at the difference in that might help us understand how they evolve these differences and also how they may use them for swimming. So that's why we measured these things. And we measured, so I'm going to show you the abbreviations for these parameters. I'll try to remind you every time, but still. The outer diameter here is big D, the inner diameter is little d, the angle is theta, and the length is CBL. Again, I'll probably remember to remind you, but just in case. I'm also going to take the time here to pass out these models so you can see them. These are representative of these species here, just a couple of them, so feel free to pass them around. If they break, don't worry about it. I printed them at low resolution, so they might break. You can take a look and see the differences in vertebrae, which are hard to see on the screen, but easier to see in 3D. Anyway, yeah, so we measured the vertebral morphology of the fish. This time we measured it in 15 different species. So here they are, all of the species. And I want a special shout out to one of my collaborators, Alexis Roberts, who came up with the idea to study non-elongate fishes in this way. I was very focused on elongate fishes because I love them the best. But she, reminded me that there are other fish in the sea that are not elongate and that we should maybe consider looking at them too. So she brought the lingcod and the sculpin and the surf perch and the bluegill all into the mixture here, so we have a nice model with lots of differently shaped fishes. So thank you, Alexis. So we measured those vertebral parameters in all of these species along the length of the body, many individuals of each species. There's a whole thing. And what it looks like when we plot it all out, so this is position, again, on the x-axis, and this time on the y-axis I'm showing you the canal diameter, so the little tiny bit in the middle. And this is what it looks like for all 15 species, the canal diameter, for one thing. So this was a lot. It's really cool to get lots of data, but it makes it a little bit hard to analyze. And when I first got to Tufts, my understanding of how statistics worked was not a thing. So my first thought, of course, when I saw piles of data, why don't I just compare everything to everything? It seems like a great way to go. If you have all of your variables of interest down the middle here and you cross them with all the other variables of interest, you can see stuff. The problem with this method is, one, it's ugly and boring, and two, the vertebrae aren't individual things. So D is not separate from body length or angle or small diameter, it's one unit. They all work together to create a vertebrae and to create movement. So separating them out and looking at them one piece at a time doesn't really make sense. So thanks to my committee members and also Eric Scott, I learned the power of multivariate statistics. And the method that we chose to use for this is partial least squares regression, which, short story of partial least squares regression is it takes all of those measurements that I showed you before, of all the vertebrae and all the places, and it reprojects those data onto axes that best describe the thing we're interested in. So in our case, wobble, torsional stiffness, bending amplitude, but in theory, any sort of swimming thing that we're interested in. We did have one other problem that we had to jump over. We had this position variable, Like I've been showing you over and over, like you always have everything against position along the body. In this case, I'm showing you the tone angle and the body length of these three species of fish here. And if we put position in our model as just another variable, we run into the problem of it varies with everything. So everything relates to position. So we couldn't really just add it in there because then the only thing that would come out as being important would be position. And it would ignore the rest of everything else. So the way we solved that problem, this was again Eric Scott's brilliant idea, was to fit quadratic lines to the body. So you've been seeing these lines that I've been putting in on all these plots. All of the morphological parameters that I've been showing you have to do with, or vary along the length of the body in a kind of quadratic way. So there's kind of a curve, which makes it really nice because we can put one of these on each one of the things we measure. That being said, I want to show you what the quadratic curve means because we actually use the coefficients of those curves in the model. So brief back to early math on what a quadratic equation is. It's described by ax squared plus bx plus c, and the coefficients that we used for our model are a, b, and c. So we ended up fitting a line to the model, getting those coefficients, a, b, and c, and then putting those into the PLS model instead of putting the actual vertebral measurements. So if you think of this x-axis again as position along the body of something, we can think about how varying these coefficients would vary morphology along the length of the body. So if we increase A, for example, the curviness of our line increases. So it went from a nice smooth curve like this to a really sharp curve. If we increase B, the slope changes. So instead of being sloped down like this, the curve moves up. And finally, if we change C, the entire curve shifts up. So that's pretty much the mean of the data shifts up. So we put all these curves on all the data, put them in PLS, and ask the question, how does vertebral morphology affect bending amplitude, wobble, and torsional stiffness? Those are the things that I've been looking at. Cost in bending amplitude, I'm not sure if I explained that, but bending amplitude is basically the amount that the tail goes back and forth when the fish is swimming. And we actually measured amplitude down the length of the body. So here there's a small amplitude, here there's a bigger amplitude. We have those data for all the fish as well. And I'm going to show you some of the results for bending amplitude first. So here's a bunch more plots of position on the x-axis, which you're used to seeing by now. And this time I'm showing you bending amplitude on the y-axis. And I'm going to show you a few plots that look like this. So this is the same gunnel I've been showing you all along, my favorite fish. The green line is what the model predicted that the bending amplitude would look like in this fish. And the blue line is the actual bending amplitude that we measured in the fish, just in the field. We took videos and measured bending amplitude. And for this fish at the top, and for a couple other near-shore fishes, that's what the red lines represent here, our model does a pretty good job. We can see that we can pretty accurately predict bending amplitude in near-shore fishes along the length of the body as they're swimming. We also tried to do it in other groups of fishes. So the bottom row here is subtidal fishes. And we can see that it does slightly less good. Our means are all over the place, so we're either predicting really high or predicting really low. But we're getting close with the slopes. So what I think the reason is for the means being off here is that the model is pretty heavily skewed right now towards elongate fishes, seeing as I like elongate fishes. So most of the fish in the model are pretty bendy, flexible, these guys. So we're hoping that if next time we run this, if we get more normal shaped fish, it might help us even out some of those means. But instead of showing you a plot like this for every species, I'm going to show you a quick summary of what all of that looks like. And to do that, we're looking at root mean squared errors. So that's basically going to show you the difference of the predicted line and the fit line. So a really high difference would be a high root mean squared error, and a really low difference at the end of this here would be a low root mean squared error. Just to give you an idea. All right, so for bending amplitude over here, we found that when we broke up everybody into their habitats, our model did a pretty good job. So you can see here there's some variation, but the model did a really good job predicting bending amplitude really well in nearshore fishes, and benthopelagic fishes. And the reason I think that is because nearshore fish and benthopelagic fish are pretty specialized. The nearshore fish, like I said, are always following the tide in and out, so they have a thing they have to do every day. And benthopelagic fish are really specialized because they tend to just hover along at the bottom of the sea, so they don't really sink and they don't really float. They're really good at hovering and hunting that way. So to be a really specialized predator, you have to have specialized morphology. So to me it makes sense that their morphology would predict their behavior better than the slightly more diverse fishes in the other groups. We also used the model to answer some questions about torsional stiffness and wobble. We only had 6 species in both of those groups because I only measured 6 species in my first study. But we found similar results. If we tried to do the model with all 6 species at once, we got a lot of error. And if I took just the benthic species, so in this case, benthic fish just stay at the bottom. They're pretty much always on the bottom of the ocean floor. They enjoy dragging themselves around down there and eating food off the ground. So if we looked at just the benthic species, it did a really good job. Similar with wobble. It was kind of a big spread of error if we looked at all 6 species, but if we looked at just benthic species, the spread went down. Cool. So, I think I can say from this that vertebral morphology seems to be a pretty good predictor of swimming mechanics, but what's next? How else can we use this model for anything else besides saying, yes, I can tell you how a fish that I know how it swims swims? It's an important thing that we got out of this model was taking all these fish that I showed you before and breaking them up into their habitats was a really good way to figure out their kinematics. So looking at their ecology turned out to be really important. You couldn't just ignore the fact that they are from different places. Even if they're related, if they live in a different spot, they probably have different morphology. And that turned out to be really important. So we can also use the model, the results from it, the weights of the model, to tell us how these different groups evolve differently to accomplish the same task. So if we look at the coefficients of those lines that I was showing you before, the different coefficients lead to different morphologies. So that tells us how each different group evolved their vertebrae differently to live in the same or different environments. So I'm going to give you an example here. Say all of our groups, the intertidal, nearshore, and subtidal groups, wanted to decrease bending amplitude. And wanted to is a strong word here. I mean, they evolved to decrease bending amplitude. They can't just change their vertebrae when they feel like it. So to decrease bending amplitude, these 3 groups achieved it by doing a different thing. So intertidal fish achieved a decrease bending amplitude by stretching out their vertebrae, increasing the diameter, and then changing the diameter from head to tail. So that is how intertidal fish use their morphology to decrease bending amplitude. For nearshore fish, they do something a little different. They stretch out their vertebrae just like the first one, but this time they change the angle of their vertebrae, so they change this cone angle down the length, and they increase the canal diameter down the length of the body. And finally, for subtidal fishes, they kind of just keep everything the same. You'll notice that all these are negative, which means they don't change a lot of things. They like to have similar vertebrae down the length, and they decrease the mean diameter a lot, so their vertebrae are just thicker. They just like to be stiff and annoying fish. So it's kind of interesting. We can take the results from this model and make some predictions about how these animals evolved to do a thing. Another cool thing that I've been hinting at throughout is being able to use data like this to try to make predictions about how fish swim when we don't actually know the answers. I've known the answers to all of the swimming questions before, and I've just tried to compare them, but this is a case where we don't know the answer. So this fish here is another intertidal fish that we didn't actually measure swimming kinematics for, but we have its vertebral measurements. So if we use the measurements of vertebral morphology across the length of the body and plug it into our model, our prediction for how his bending would look along the length of the body looks something like this. And to put that in context, we have 3 other species that are related and live in the same area, still intertidal fish, and they fall out in a pretty similar spot. So this guy is the predicted fish, and these 3 are the actual values of related fish. And they fall out in a really similar area, which is what we would expect. We would expect this fish to swim similarly to all these fish. Just to try it out again, we looked at nearshore fish. This time we chose two fish that look kind of different. So one of your more standard elongate gunly looking fish. It's not a gunnel, I know, but it looks like one. And a little bit more stiffer tube snouty fish. And we can see that the model predicts different amplitudes for the different fish. The longer, bendier guy has a higher amplitude, and the slightly less long, less bendy guy has a smaller amplitude. And if we look at the other fish in this group, it falls out again in a somewhat predictable way. These two lines here, from the fish that we know the answer to, are these two other gunly-looking fish. So this fish swims similar to this fish. And this line is that sand lance up there. We'd expect him to bend less based on his morphology too. And the tube snout is falling closer to that fish. So I would say it does a fairly good job at making some predictions about how fish move. So, does the vertebral morphology affect body and swimming mechanics? I would say yes, and we can use it as a thing to make predictions, which is pretty cool. So I just wanted to summarize what I've gone through today, the chapters that I went through. First, we looked at wobble and found that wobble in different habitats, fish in different habitats wobble differently. It's more important to look at their habitat than it is to look at their family relationships or their size, shape, etc. We also looked at torsional modulus and found that wobble is not related to torsional modulus, so it's not just the mechanics of the body that predict how fish swim. There's other things to look at. And those other things could be vertebral morphology, where the data from this actually ended up predicting swimming kinematics a lot better than torsional modulus. And we can hopefully use models like this. I really hope that people take this and run with it. We can use big inclusive models like this to make predictions about other fishes that we can't really measure swimming for. So deep sea fishes, where the only view you'll ever get of them is briefly in the camera and then they'll run away. Extinct fishes, anything else that's hard to keep in the lab. We can use models like this with fishes that we understand to make more educated guess as to how these fish might have swim and how they would behave in nature. And hopefully we can then start applying studies like this to all sorts of different types of fish swimming. I know what I was looking at today is mostly just body caudal swimming, so bending down the length. But it'd be really cool to try to apply a similar study to pectoral fin swimming or weird tail flapping like the other guy does. Yeah, so I have to thank people now. That's cool. Alright, so I'm sorry if I forget anybody in advance. First, my advisor, I'd like to thank Eric. Eric is probably definitely the best advisor ever. The rest of you all can come at me. Yeah, I've never worked for anyone like as patient and understanding of my insanity as Eric is. A lot of the times, like my favorite description of how Eric interacts with us is I'll go into his office and be like, hey Eric, I want to do this thing, and he'll be like, I think that's a good idea. And I'm like, okay, but I've already signed up to do it. And he was mostly like, fine. Let me know if I can help. Like he's always, even if I do the thing that he told me not to do, he'd still get my back and help get me through it. So that was really awesome because mostly he was right. If he said doing the thing was a bad idea, it was a bad idea, but I did it anyway. I'm a terrible listener is the short story. And Eric was really great at dealing with that. The rest of my committee I'd like to thank as well. Barry, thank you so much for just unlocking the door to your lab and being like, have fun in there. That's basically what he did when I got here. He's like, yeah, Cassandra, sure, just here's everything. Use all the tools, make soft robots, it'll be great. So I really appreciate you just letting me have access to everything and letting me be on the team. Elizabeth Crone, teaching me everything I know about statistics. I don't know where you are in the audience right now. There you are. Like I said, I came in here having never taken a stats course or knowing what it was or how useful and cool it could be. So Elizabeth taught me all of that. And I really appreciate it. I feel like I can leave here knowing like how to do actual science, which is cool. And then last but not least, Adam Summers, who is in the computer in theory maybe. And Adam is the PI out at Friday Harbor, where all these fish came from. So he was in charge of making sure I didn't do anything stupid while I was out there. And when I say in charge of making sure I didn't do anything stupid, I mean encouraging me to do stupid things for the entire summer. And also making me fix all of the lab equipment as it breaks. So thanks, Adam. I'd like to thank the lab. Here's the lab. Especially Mike, who dealt with my bullshit for a long time. I don't know, parents, do you remember how before every exam in high school I would panic? It's like that, except for 5 years. Mike had to deal with all that, and he was really good at it. He took me for coffee walks and was like, I feel like you're spiraling here, Cassandra. So I really appreciate that, because I definitely needed someone to tell me that I was being crazy to help lessen the crazy. Yanni is here too, and I'm very excited that Yanni is here. Hi, Yanni. Yanni also sat there and listened to me whine about things constantly. And Kelsey, I need to thank multiple times, because she listened to my talk and also kept me excited about stuff on Twitter for leading up to this whole thing. Rest of the lab, I'm sorry if I forgot you all. Caitlin and James, our senior undergrads, have been awesome. And I've worked with a lot of really, really awesome students and undergrads and master's students here at Tufts, and they've made all of this data possible. I have to thank my collaborators. Specific shout out to Marianne, who introduced me to fish. I had never studied fish before, and she was like, you just study fish, they're cool. Also, you should study vertebrae, they're cool. Here we are. She also convinced me that it wasn't scary to email Eric. Because when I was applying to grad school as a engineering major or whatever I was like, I can't email a biologist, they're never going to accept me. She's like, you'll be fine. So thanks, Marianne. And then other collaborators, Eric, thank you so much for all of your help with programming, stats, the art, and everything. Alexis for giving me the idea to look at not just elongate fishes, look at everyone else. And she's collected so much data over the summer. It's a dissertation's worth of data in 2 weeks. It's insane. And Sarah and Matt both helped with keeping me sane. A lot of these things are keeping me sane, if you notice it. And Matt also called me on the phone and went through one of my chapters with me when I didn't know words anymore. So I appreciate that. Sorry, this is going to keep going. Can't get away with this. It'll be over soon. So first, FHL crew. These are all the people that run the labs, are at the labs, or just generally present at Friday Harbor when I am. And they pretty much make the place what it is. It's not the same without any of them. They're great. Dorm Q et al. Are the UW grad students that I hang out with every summer. They're awesome. We all do crazy things together and keep each other alive. It's wonderful. The Tufts admins for keeping the department alive, because I don't know what we would do without you guys. It would fall apart instantaneously. And also my cohort. Hello, cohort. My cohort is great. We do fun things together. And I super appreciate all of you guys. One more. Okay. Special thanks to my office mates, because, like I said, sharing an office with me this summer probably wasn't the easiest thing ever. But I really appreciate you guys for chatting with me and being patient, and especially Ruby for working there with me every weekend. And just, yeah, we should get some sleep ever. Thanks, Ruby. Other Tufts people who I hang out with a lot, I'm sorry if I forgot people, but you guys are all awesome. You know who you are. I appreciate you. These folks all listened to my talk, and this is not all of them. The rest of the lab listened to my talk, but I didn't want to put everyone's names on there 18 times. Some of my not science friends who I've managed to keep through grad school, which is a struggle, let me tell you. I super appreciate them and their support as well. Finally, one more thing. My family. Here they are. My parents are there, and my brother is here. This is a photo of them looking all nice and summery. My parents and family are awesome. They've always been really supportive of all the insanity and putting up with me not calling them for weeks when I'm stressed out, or calling them every day when I'm stressed out. You never really know. And I've got a lot of cousins and uncles and aunts, and they're all great. This is my favorite picture of me and my cousins. I don't know if you can see it, but it pretty much describes how my family and I are. We all hate pictures equally. I'm just saying. Last but not least, I want to thank you guys and also all the fish, and that's it.
You did it.
Aw, you guys. This is supposed to be I can't tell if you're waving fish flags or asking questions. Yeah, okay. I'll come over here.
Okay.
Stuff like this.
Thank you. That was lovely. I have two questions. How is wobble different from a roll? And can you comment on what the muscles do during the swimming? Because I know you've accounted for vertebral quality and you've talked about torsional stiffness. Maybe the error that you're getting is because of some backing of the muscles that are different in these fishes along the leg.
Yeah, that's a super good point. I actually originally, way back in the olden days when I was proposing, I'm like, I'll look at muscle too. Don't have time for that nonsense. Answer your first question, how it's different from a roll. A roll is when the whole fish's body just flops over to one side or the other, so if it gets hit by a wave or if it just kind of dives to the side, its whole body rolls. And wobble is more of a twisting along the length that's happening during swimming.
So it's roll just with dx across the leg.
Yeah, exactly. It's the same axis though. And yeah, no, we didn't do anything accounting for muscle here. We tried to account for body shape by looking at the cross-section in all of our things. And since fish are basically just muscle tubes, looking at the cross-section basically is looking at muscle mass. It'd be really interesting to see how the muscle could control wobble. The muscle that would do it is really, really tiny and just down the middle. And we'd have to make, I think, a physical model to test that out before we tried to implant EMGs in tiny fish muscles. But that originally was the thing I wanted to try, and maybe it's something we could do next.
This is something maybe I should have asked you earlier, but I noticed in the the vertebrae that all the measurements you're making are just on one side of the vertebrae, but they're not symmetrical. And so I'm curious if there's variation in that other part, or if there's a reason why you only have the diameter on the one side and the angle on the one side.
That is a really good question. Mostly, my short answer is we picked a side for simplicity.
Sure.
I picked the side that had the largest open part, so the part that would have the most soft tissue in it. For some fish, it's pretty symmetrical, like one of the ones you've been passing around, you'll see that both cones are the same. And for the gunnels in particular, they tend to have a really big cone posterior facing and a small cone anterior facing. So we just stuck with measuring the posterior facing cone in all of the fishes. But it would be really interesting actually to look at the variation front to back. That might add a lot to the model actually. But yes, I did notice that.
So I thought it was really cool how you could take the change in predict how they might evolve those different habitat types in the states. But do you have any sense of how easy it is to change those? I guess what I'm wondering is how many genetic elements are going in? Do you have a sense of how many genetic elements it requires to create those structures?
No. I'm gonna go with no. I'm not a geneticist and I'm crap at genetics. But I know that these changes happen in small increments over time. But if we look at vertebral variation across all fish, it's actually the thing that seems to vary the least. So that cone, that double cone shape that all fish have it at the top of the tree, fish have it at the bottom of the tree. The only thing that's really changing is the extent of, or the angle of the cone, or the width, or just very little things. So I imagine that it's something that's conserved over evolutionary time, and just the little tweaks might be what's helping. But no, I don't know what genetic factors go into changing vertebrae, unfortunately. Sorry.
Kind of seems to me like there might not be a lot of difference genetically, but their environment when they're growing up could be a big deal. So you might want to challenge yourself with collaborator going into that. But if you could probably compare like wild caught to like bred in a tank, because I think kind of like their environment as they're raising can have a big effect on their
Yeah, that's actually a thing that's been looked at before. How you raise the fish actually does change the way its bones and vertebrae develop. So there have been studies raising fish in water and on land at the same time, or different fish, but same individuals in water and on land. And the fish that are raised on land will have thicker, denser bones than fish raised in the water. So yeah, their development over the course of their little larval lives to their adult lives has a lot to do with the change in morphology. So that would be a cool thing to look at. Thanks.
Well, if there are no more questions, let's say thanks again. So the committee will probably have a few questions for Cassandra, but assuming she satisfies us with her answers, there will be a celebration at 3:30 outside the bio main office. If she doesn't satisfy us, we'll still have a celebration, but we'll see how that goes. So come back at 3:30 and have some champagne.
Thanks, Ben. How'd it go?
Good. Let me turn it off.